Thank you, Rick. Good morning, good afternoon, good evening. First of all, I would like to thank Active Motive, not only for inviting me to present our work today, but also for co-organizing with me the Chromatin Club Miami, which actually is designed to give more visibility to young PIs. And I think actually it turned out to be quite popular. So the overarching goal of my lab is to define epigenetic dependencies in cancer with a big aim of identification of new therapeutic opportunities. Today, I'm going to talk about some work that we are doing on PRC1 in Polycomb repressive complex 1 and CoREST. And interestingly, it's actually the non-canonical role of PRC1. So it's going to be gene activation and also the role of CoREST in gene activation in metastatic breast cancer and cell plasticity. So 1 in 8 women actually will develop breast cancer in the US in 2021. Breast cancer is the most common cancer in American women. All breast cancers arise in the terminal duct lobular units, which are the functional units of the breast. And the classification of breast cancer subtypes on the basis of molecular and histological characteristics have been developed. Here you can see luminal A, luminal B with accounts for 70 to 80% of all breast cancers. They express estrogen and progesterone receptors. They are low-grade tumors, and patients are treated with chemo and also hormone therapy. Other breast cancer subtypes have moderate or high levels of another receptor called HER2. These subtypes are more aggressive than luminal, but still can be treated with targeted therapy, named anti-HER2 therapy, and more recently also with PD-L1 therapy. The most aggressive subtype of breast cancer is the triple negative breast cancer, which does not express any of these receptors. And this is, as I said, is very aggressive, and there are very limited treatment options, being chemotherapy the standard of care. So we're going to talk a lot about ER-positive breast cancer today. So here is how basically the estrogen actually regulates gene transcription. It binds to the estrogen receptor, which dimerizes, goes into the nuclei, is recruited to the estrogen responsive elements, which leads to recruitment of co-activators and therefore gene transcription. So how patients are treated usually with endocrine therapies, and there are 3 main therapies. You can have aromatase inhibitors, which basically block the production of estrogen, and therefore genes cannot be activated. Estrogen responsive genes cannot be activated. You can modulate the estrogen response by adding tamoxifen, which competes with estrogen, and when it binds to the receptor, recruits co-repressor to repress gene transcription. The last example would be the degraders, being fulvestrant the most famous one. And what it does is basically impair the mobility of the estrogen receptor and leading to basically degradation, and therefore, of course, no gene transcription. So despite success of endocrine therapy, resistance arises in 40% of patients. Therefore, there is a critical need to identify new therapeutic targets. So my lab has been focused about Polycomb complexes, as Christian Helin just explained. You have PRC1, which deposits to a ubiquitin, and you can have multiple flavors, dozens of PRC1 complexes that can be assembled in a given cell. Then you have PRC2 with ECH1, ECH2, which deposit H3K27 mono, di, and tri, as he beautifully explained a couple of hours ago. And also, we don't really need to discuss a lot about this, because the canonical function is basically that both complexes cooperate to maintain gene transcription. And there's a lot of work done by many labs trying to understand how these Polycomb complexes work together. So during my postdoctoral studies, I studied PRC1 in stem cells. We found that CBX proteins have specific roles in stem cell pluripotency and differentiation. We characterized some canonical and non-canonical PRC1 complexes. Also, we found that PCGF2, which is a specific canonical PRC1 actually. Subunit regulates cardiac differentiation. And when I set up my lab, I said, I don't want to study more polycomb and stem cells because there are many labs out there doing excellent job. And I said, okay, what is the role of PRC1 in cancer? Because as Christian pointed out before, PRC1 genes are non-mutated in cancer. And actually, if you go to cBio portal and you check what is the alteration of PRC1 genes in cancer, you will see that they are not mutated, but they are amplified. And they are amplified in many different cancer types. And you can see here in breast, 3 different data sets show that at least one PRC1 gene is amplified in breast cancer. So we decided to study breast because I said there are 3 data sets. So we thought that the data was robust enough to invest some time and years on that. So RNF2 is the gene that encodes for RIN1B, which is the E3 ligase, and in principle deposits monoubiquitination on histone H2A on lysine 119. You can see that amplification of the genes correlates with overexpression, specifically in luminal breast cancer, ER-positive breast cancer. So the first thing that we did was just to map where does RIN1B bind, right? I mean, we know when we did this as a control, we use human IPSs where we found, as expected, that RIN1B co-localizes with PRC2, H3K27 deposited mark, and also H2A ubiquity, right? The surprise was when we did RIN1B ChIP-seq in non-tumorigenic breast cells, MCF7, or triple negative breast cancer, or ER-positive breast cancer cells, we found that actually RIN1B mainly binds to K27 and H2A ubiquitin-free sites, right? Only 30%, 25, or 20% of RIN1B targets actually contain H2A ubiquitin and K27 trimethylation, suggesting that RIN1B and PRC1 might play a non-canonical function in more differentiated cells and in cancer cells. Indeed, here you can see that RIN1B is recruited to super enhancers. Here, the super enhancers that regulates FOXA1 in ER-positive breast cancer. And you can see that there's a nice co-localization with the estrogen receptor. So we went to other cancer types, and we found that this localization of RIN1B to active enhancers is not only happening in ER-positive breast cancer, but also in triple negative, in leukemia, and hepatocellular carcinoma, where we see co-recruitment of PRC1 with ER, or BRD4, or GATA1, etc. I am not saying that PRC1 does not repress genes in cancer, in breast cancer. Here you can see triple negative breast cancer, which do not express FOXA1. And you can see that RIN1B is recruited to FOXA1 to repress and deposit H2A ubiquitin. But what is clear is that PRC1 can have a dual function in breast cancer as a repressor and as an activator of transcription. So in this paper published a few years back, we didn't show a lot of mechanisms, so we decided to investigate what is the role of RIN1B in the estrogen pathway. So as I said, estrogen binds to the receptor, dimerizes, and is recruited to estrogen-responsive elements, where recruits co-activators. So how can we study PRC1 and the estrogen pathway? We can culture ER-positive breast cancer cells in hormone-deprived media that does not contain estrogen, and we can induce these cells with very low doses of estrogen for 45 minutes, 4 hours, 8 hours, 12 hours, or 24 hours to investigate changes in gene activity, chromatin accessibility, genome architecture, etc., etc. So the first thing we did is we depleted RIN1B, and we give cells estrogen, and we did RNA-seq at different time points. And you can see that around 900 genes become dynamically upregulated during estrogen administration. And you can see that when you deplete RIN1B, this estrogen-induced gene expression is strongly attenuated. Again, indicating that RIN1B may play a role in gene activation here. Indeed, you can see that H2A ubiquitin does not change when we deplete RIN1B, again, supporting the notion that RIN1B is not the main E3 ligase in this in breast cancer and in ER-positive breast cancer. But what we can see is that actually chromatin recruitment of RIN1B is essential, because when we did the rescue of this of the knockdown cells with the mutant that cannot bind to chromatin, we cannot basically rescue the estrogen responsiveness. With the wild type, we rescue 100%, of course. With the mutant that cannot bind to chromatin, nothing. But you can see that when we rescue with the catalytic dead mutant, we can partially rescue, suggesting that there is a non-histone substrate of RIN1B in these cells that we have not yet, unfortunately, been able to characterize. You can see here that when you give estrogen, there is an accumulation of H3K27 trimethylation, which is attenuated in the RIN1B knockdown. So we ask, well, maybe RIN1B, it's important for chromatin accessibility. And of course, we did a TAXIC. And you can see in these time points, as you can see here, an example in the genome browser, GREB1 is a bona fide estrogen responsive genes. You can see that the promoter is opening when you give estrogen, but you can see that in cells like in RIN1B, you don't have a chromatin opening, and we can see this basically genome-wide, indicating that RIN1B regulates estrogen-mediated chromatin accessibility. This lack of accessibility results in less H3K27 acetylation and enhancers and promoters, as it was suggested in the Western blot that I showed in the previous slide. One of the main questions in the polycomb field is how polycomb is recruited to chromatin. What are the mechanisms that induce PRC1 or PRC2 recruitment? And of course, we hypothesized that estrogen would be a candidate. Therefore, we did RIN1B to test it. We did RIN1B ChIP-seq minus plus 45 minutes of estrogen, along with ER alpha, ChIP-seq, and FOXA1. FOXA1 is the pioneer factor of ER, so you need to have FOXA1 before estrogen administration in order to have ER alpha recruitment. You can see that it's very clear that estrogen induces RIN1B recruitment at sites that contain the estrogen receptor. Depletion of FOXA1, I'm not showing here, abrogates recruitment of both RIN1B and RIN1A. Interestingly, this recruitment of RIN1B upon 45 minutes of estrogen is dynamic, because you can see that after 8 hours of estrogen, there is a strong release of RIN1B from chromatin, which then comes back after 24 hours. And if we do ER alpha ChIP-seq in the control and in the knockdown RIN1B cells, you can see that RIN1B is required for late estrogen recruitment, but not for the early estrogen recruitment. The lack of estrogen recruitment here also results, as expected based on the previous results that I showed, in less accumulation of H3K27 acetylation, and therefore no expression of TFF1. In our efforts to identify the RIN1B interactome here, of course, we did RIN1B proteomics, IP, pull down, and then mass spec, and we only found canonical PRC1 genes. We didn't find the estrogen receptor, which was a little bit surprising based on the ChIP-seq data that I've been showing you until now. So we decided to do single nucleotide resolution binding profiles of ER, RIN1B, and other cofactors. And you can see that here, this is the classical profile of ChIP-seq, where you have minus plus estrogen. This is recruitment of ER, minus plus estrogen. This is the recruitment of RIN1B. You see nice overlapping. If you do ChIP-exo, which uses an exonuclease that degrades 5' 3' DNA, you can see a very nice profile here, where you can see that ER should bind here between the summit of both peaks. This is where the footprint is of ER alpha. You can see these ChIP-seq peaks is actually 2 ChIP-exo peaks, and the same with RIN1B. I guess you can see that they are not actually on top of each other. So what we did is that we center ER alpha ChIP-exo peaks, and we asked, where is RIN1B binding genome-wide? And actually, we can tell that it's literally binding 10 nucleotides to the right of ER. Other factors like RNA-2 is binding to the left of ER. And FOXA1, it doesn't really matter, because as I said, FOXA1 is recruited before ER at chromatin, right? So Now it's not surprising that ER and PRC1 or RIN1B, because we did this with other PRC1 subunits, they don't interact because actually PRC1 is recruited to the right of ER alpha. But I just showed that RIN1B is recruited with ER, right, in the presence of estrogen. This is the grade 1 promoter. This is the proximal enhancer, where you can see co-recruitment of RIN1B and ER. And you can see distal enhancer here, which is strongly activated after 45 minutes of estrogen, also contains ER alpha and RIN1B. So we asked whether RIN1B was important for enhancer-promoter interaction to activate gene transcription. So again, I want you to remember that here we're talking about RIN1B in gene activation, which is completely the opposite as what you would expect from a polycomb gene. So we did 4cc, and you can see that control cells 0 and 45 minutes of estrogen. There is a strong interaction of the distal enhancer with the promoter. We use the promoter as a viewpoint. You can see when you deplete RIN1B, this enhancer promoter interaction is completely lost, and we did nascent RNA, and of course the gene is not transcribed. So this is one of the few examples, the first example of RIN1B being important for enhancer promoter interaction during estrogen administration. So it is known that, going back a little bit more in the mechanism, it is known that R loops are formed during gene transcription, right? And they've been suggested to be also recruitment platforms. So R loops are 3-stranded structures that harbor RNA-DNA hybrid. And we ask, first of all, do we see an accumulation of R loops after 45 minutes of estrogen? And you can see here in light blue that you have a strong accumulation of these R loops at sites that contain ER and RIN1B after 45 minutes of estrogen. When you deplete RIN1B, you can see that there is less R loop formation, genome-wide at the co-targets of ER and RIN1B, and this is one example. So here we have a link of RIN1B with R loops. We did EMSA assays to see whether RIN1B binds to this structure, and it's not binding to R loops. So we still don't know the mechanism here, but what we see, and here we copied an experiment performed by John Reed in a beautiful Nature Genetics paper published last year, I believe it was last year, where he did, he asked whether PRC2 needs RNA to be stabilized at chromatin. So we asked the same question. So we did ChIP-seq of RIN1B and ER in the presence of RNase A, and as expected, ER alpha doesn't need RNA to be stabilized at chromatin, which is in line with the recent Cell paper from Dr. Davide Ruggero. But interestingly, we saw that RIN1B actually stabilization at chromatin requires RNA. Here you can see that genome-wide ER alpha is not affected when you treat the chromatin with RNase A. RIN1B is 50% destabilized from chromatin. Here you can see. So we still don't know exactly what are the RNA molecules that affect this RIN1B stabilization at chromatin. We're working on this. We're doing prime and capture proteomics to try to address these questions. So to summarize the first half of the talk, I told you that estrogen induces PRC1 recruitment to enhance and promoter interactions, and this recruitment is important for enhancer promoter loop, ER alpha recruitment, chromatin opening, R loops, and transcription. So the second half of the talk is about a non-canonical role of a CoREST complex in metastatic breast cancer and cell plasticity. So as I pointed out in the introduction, patients with luminal breast cancer, ER-positive breast cancer, they receive endocrine therapies, which could be tamoxifen, fulvestrant, or aromatase inhibitors. And even though this treatment really works, after 5 to 10 years, 30 to 40% of patients unfortunately relapse. And the mechanisms of resistance are actually quite diverse in breast cancer, because you can acquire mutations in the estrogen receptor, in MAP kinase, in the SWI/SNF complex, FOXA1, but also there is an understudied mechanism, which is basically the loss of the estrogen and progesterone. This happens only in patients with mutations in P53. And basically what I'm telling you is that patients start having, they have a luminal ER positive breast cancer. After endocrine therapy, they become basal-like or ER negative breast cancer. So this is very well, this is not well studied because there is a lack of preclinical models to study this. So we wanted to first generate the first preclinical models and then to identify what are the drivers of this cancer cell reprogramming or plasticity and whether we can find new therapeutic options for this incurable breast cancer. So Liliana, Ph.D. student in the lab, took ER positive breast cancer that are wild type for P53 and also mutant for P53, sorry. And she cultured them in tamoxifen fulvestrant for over a year until they become resistant. And also in LTED conditions, also known as long-term estrogen deprivation. This cell culture condition, basically what it does is to mimic aromatase inhibitor treatment. So basically we have 6 cell lines, right? Cells that are resistant to endocrine therapy, either by culturing them with tamoxifen, fulvestrant, or LTED for over a year. And we ask whether we see luminal-to-basal reprogramming, and for this, to do this, we did CD24 and CD44 stainings as well as keratins, and also we assess whether there is an ER and PR loss. And among all these cell lines, only one of them, P53 mutant, as expected, cultured in LTED for over 1 year, fulfilled this criteria. And here I show you that CD24, this is associated with ER positive breast cancer. CD44 is more a cancer stem cell and a basal marker. And you can see that when you culture these cells with in LTED for over 9 months, you can see that all of them become CD44 positive. Also, the shape changes, suggesting that there is a reprogramming here from luminal to basal. We can keep these cells in culture for years. Actually, now they have 2 years and they are CD44 positive, very similar to triple negative breast cancer cells. Interestingly, we saw that the estrogen receptor protein level goes down when you culture these cells after 5 or 6 months in LTED. We see epithelial to mesenchymal transition also, and you can see here by icadine and by mentin. You can see hyperactivation of the MAP kinase pathway by phosphorylation of ER2 and phosphostat. And this is due to de novo mutations in KRAS and BRAF. So to do this, we did single cell SNP analysis in normal cells, parental cells, or 6 months old LTED cells. And you can see that cells in normal media, 0% of them have mutations in these MAP kinase genes. Cells after 6 months in LTED have a strong accumulation of mutations in KRAS and BRAF. And this is the RNA-seq expression levels in parental cells, full media, or LTED, reprogrammed cells, and you can see that ESR and the progesterone receptor are 100% depleted. Of course, cells in these reprogrammed cells, these LTED 9 months, they grow faster than the parental cells. And of course, they are resistant to tamoxifen and fulvestrant, basically because they don't express the estrogen receptor. We did RNA-seq during these time points. We can see 4,000 genes upregulated and 4,000 genes downregulated. Those are the upregulated. You can see that there's a dynamic change. GSEA analysis show what we expected, that now upregulated pathways are the basal breast cancer and the mesenchymal breast cancer. Also mammary stem cells, as they express CD44, and downregulation of the estrogen receptor pathways are luminal pathways. So we think that we have a preclinical model to study this cell plasticity that happens when patients receive endocrine therapy. So we have a stable cell lines with CD44, ER positive, PR. When you grow them in LTED, there is a mutation in the MAP kinase, and we don't know how and why. We don't know how, basically. After 6 months, you have like we call a prime population with a mix of CD44, CD24 positive cells. And after 9 months, you have like a basically 100% CD44 ER, PR negative. So we went back to the patient samples, RNA-seq samples from cBioportal, and we found that, and we queried what is the expression of epigenetic factors that fulfill this criteria and this criteria. And we found a lot of them, but interestingly, 3 subunits of the CoREST complex were actually overexpressed here and here. And this overexpression was associated to poor prognosis. So we decided to study the CoREST complex in more detail to see what is the role in this cellular plasticity. So what is the CoREST complex? CoREST complex is a very interesting complex because it has 2 enzymatic activities. It contains LSD1, the first histone demethylase discovered, basically demethylates H3K4 on histone H3, sorry. And also contains HDAC protein, so deacetylates proteins. It's been implicated in AML primarily, in melanoma and prostate. The role of LSD1 in breast cancer is under debate. Some people claim it's an oncogene, others tumor suppressor gene, but nevertheless, the role of the CoREST complex in endocrine resistance is actually not known. So we decided to knock out LSD1 or knock down LSD1, sorry, in the 3 stages, stable lines, 6 months old, fully reprogrammed cells, and you can see proliferation is strongly affected when you don't have LSD1. We rescue this phenotype when we reintroduce both wild type LSD1 and catalytic dead mutant, indicating that the enzymatic activity of LSD1 is not important for cell proliferation and cell fitness in this particular context. In agreement with the previous data of the rescue, we didn't see actually major changes in H3K4 dimethylation in wild type and knockout, both reprogrammed cells and parental cells. We did acid extraction histone followed by mass spec in both parental and reprogrammed wild type knockout cells, no difference at all. And actually, only 2% of H3K4 dimethylation peaks increase in the LSD1 knockouts. Suggesting again that LSD1 function here is not due to its demethylase activity. In agreement also with that, we tested whether GSK LSD1, which is a compound that targets the enzymatic activity of LSD1, would affect the proliferation of the parental, primed or reprogrammed cells, and we did not see any effect. This is an IC50 assay. Basically, they are resistant to the drug, also to TCP, which is the first actually LSD1 inhibitor, and 2 analogs of this GSK LSD1. Interestingly, Phil Cole from Harvard recently developed a compound called corin, which specifically and only targets the CoREST complex. It's a dual compound that has an HDAC inhibitor and an LSD1 inhibitor, and only works when you have HDAC1 and LSD1 in the presence of the CoREST complex. So, of course, what we did LSD1 pull down, mass spec, to first confirm that LSD1 actually interacts with the CoREST complex. And you can see here in orange, both in the parental cells and in the reprogrammed cells, LSD1, of course, interacts with all the subunits of the CoREST complex. Interestingly, we found multiple subunits of the Schweizniff complex, both in the parental and in the reprogrammed cells, and I will go back to the Schweizniff complex in the next 5 minutes. Therefore, we could use corin and test corin in our system, and yes, corin is very important. When you treat the cells with corin, the IC50 is around 400 nanomole. The cells are very sensitive to corin and they don't really proliferate. So a little bit of mechanism, we did LSD1 chip-seq during this reprogramming. This is the parental cells, prime cells, where actually we also sorted out the CD44 positive cells and the fully reprogrammed cells. So for the sake of simplicity, just focus on the first and in the last heat map. And you can see that there are specific LSD1 peaks in parental cells and in the reprogrammed cells, and some common peaks. It was very interesting because when we did the Homer analysis, we found that LSD1 binds to sites that contain binding sites for FOXA1, ESR1, estrogen receptor, and GATA3. This is the main, as I show you in the first part of the talk about PRC1. This is the key. Those are the guys that actually regulate the estrogen responsiveness, right? Interestingly, during this evolution of resistance, we can see now that LSD1 binds to AP1 sites and CTCF sites. Therefore, this leads to a very important question: is maybe actually CoREST has a different function in parental cells and in reprogrammed cells, right? So maybe in endocrine sensitive cells, the parental cells, CoREST is recruited to ER and FOXA1 sites, but then to cJUN or AP1 sites in the endocrine treatment resistances. So of course we tested that and we confirmed that. In parental cells, CoREST complex, because then we did the ChIP-seq of another CoREST subunit, actually co-localizes with ER and FOXA1, both at active sites and repressed sites, here shown by POL2 and K27 acetylation. Right? This is an example of LSD1 co-localization with ER alpha and FOXA1. I told you that the LSD1 knockout cells, the parental cells, don't really grow, and we believe this is because the estrogen receptor is actually downregulated. So we believe this is the mechanism why endocrine sensitive cells don't grow when they don't have LSD1. I have to say, and this is very interesting, in the endocrine treatment cells, we see an accumulation of cJUN, which is in line with this potential new mechanism. So we did ChIP-seq of cJUN, and again, we confirmed that in the reprogrammed cells, the CoREST complex is recruited to sites decorated with cJUN, both active and repressed. And this is an example. This is by mentin, a very important gene in metastasis, which is highly activated, expressed in the reprogrammed cells that contains cJUN, RCOR1, and LSD1, suggesting again that the CoREST complex is playing a non-canonical function here in gene activation. So of course, we went further to investigate this potential cJUN CoREST axis. So we did LSD1 ChIP-seq in the wild type and in the knockout cells. So the ChIP-seq is specific for LSD1. Notably, cJUN recruitment was completely impaired when cells have a loss of LSD1. We did the reverse experiment. We depleted cJUN and we did ChIP-seq of cJUN. The signal is specific. And then we asked, what is the LSD1 recruitment as cJUN targets when there is no cJUN? And basically the recruitment is 50% impaired. Here's an example. Here you can see LSD1 is recruited to AXL gene. Again, a very important metastatic gene. You can see LSD1 here. Signal is lost in the LSD1 knockout. Of course, you can see here co-localization with RCOR1 and cJUN. This cJUN binding is specific because it disappears in the knockdown. And you can see that when you don't have cJUN, LSD1 also is not recruited here, but here is still recruited basically because cJUN is not recruited. Also, when you do cJUN in the knockout of LSD1, there's no recruitment of cJUN. So basically, this is telling you that there is a codependency between LSD1 and cJUN. I told you a few minutes ago that in our pull downs, we found very surprisingly some members of the BAF complex, of the Schweizniff complex, right? I'm not saying that LSD1 binds directly to the complex because I think nobody reported that. I know that this interaction is DNA independent, but it could be that there are, of course, proteins that breach this interaction. And of course, we learned a few hours ago that the canonical function of the BAF complex is to open chromatin, right? So the first thing that we did was to confirm that the CoREST complex actually is recruited to enhancers and promoters decorated with the BAF complex. In that case, with the ChIP-seq for SMART-CC1. Again, I'm not saying that these two guys interact, but definitely they are together at chromatin with cJUN. Because there is this association of CoREST with the BAF complex, of course, we did a TAXIC to assess chromatin accessibility changes. So as expected, this is the canonical function of CoREST. When you don't have LSD1, you open up the chromatin, 12,000 peaks are gained, TAXIC peaks. But also you can see that actually more than double of the peaks are from 12,000 to actually almost 30,000 peaks are lost in the LSD1 knockout cells. If you compare this profile to the parental cells, the wild type cells, you can see that these sites are closed in the parental cells as expected, and they are AP1 sites. In contrast, these sites that are open in the reprogrammed cells when you don't have LSD1 are open in the parental cells are FOXA1 sites. So basically this result, what it's telling us is that when you don't have LSD1, the chromatin accessibility from reprogrammed cells is very similar to the chromatin state of the stable cells. We decided to focus more our attention on this, right? Because this is a non-canonical function, potential non-canonical function of the CoREST complex, an association with cJUN and BAF. So you can see here an example of LSD1 and ECOR1, where the chromatin is open. When you have a knockout of LSD1, the chromatin is shut down, is repressed or closed. This also happened with the knockdown cells, so it's not an artifact of the knockouts. We can rescue this with the wild type LSD1, both the wild type mutant, the wild type version, sorry, and the catalytic dead mutant. Therefore, again, the enzymatic activity is not important for this chromatin accessibility. Also, as you can see here, by H3K4 changes that actually not only go down, up, they don't go up, actually they go a little down, which we don't understand why. And you can see less acetylation on lysine 27 and less K4. Of course, these sites that are open and contain LSD1 and ECOR1 have cJUN and SMART-CC1. The chromatin is closed at all these sites, as we show here, genome-wide, and cJUN recruitment is completely lost. We couldn't do ChIP-seq, or we didn't do ChIP-seq of the SMART-CC1 in this setting because the knockout of LSD1 induces 50% reduction of SMART-CC1. And also we didn't do the opposite experiment, that is to do the LSD1 chip in the SMART-CC1 knockdown, because interestingly, depletion of SMART-CC1 actually degrades the CoREST complex, as you can see here by a strong degradation of LSD1 and ECOR1. Right, so it seems that the CoREST complex here works with cJUN and SMART-CC1 to open, or the BAF complex to open chromatin. We took, of course, the co-bounding genes of CoREST, cJUN, and SMART-CC1, and we asked, we did RNA-seq in the knockouts and in the wild-type cells. Downregulated pathways are associated to basically cancer, breast carcinoma, EMT, MYC targets, and the upregulated pathways are associated to apoptosis, suggesting that LSD1 knockout cells should be less aggressive than the wild-type reprogrammed cells. And in fact, this is the case. We inject in the mammary fat pad reprogrammed wild-type and LSD1 cells, and we check primary tumor formation, significantly reduced when you don't have LSD1, metastasis completely impaired, and increased survival. Importantly, we repeat the experiment, treating the mice with corin, and you can see that tumor formation is actually impaired. So this is beautiful, but is this really happening in patients? Is this preclinical model that we found or generated in the lab resembles what happened in patients? So we were able to do an LSD1 ChIP-seq from a fresh tumor isolated from a patient that was treated 5 years with tamoxifen and had metastatic disease. we found 2,000 genes from this tumor. Notably, only 45 LSD1 targets were found in the parental cells, but almost 800 genes or 729 genes, LSD1 targets, were also found in the reprogrammed cells, indicating or suggesting that our cell model actually resembles what happened in vivo in patients. And this is the track of LSD1 in this gene in the parental cell or in the reprogrammed cell. You see this nice recruitment or binding, which is very similar to what we see in the patient. Importantly, when you take these co-bound sites are AP1 sites. So this was very nice to see. And just to finish, what we did now is we took RNA-seq from 21 patients that were treated with chemo-endocrine therapy, and they developed resistance. So this RNA-seq is before treatment and after treatment. So before resistance, after resistance. And from the 729 LSD1 genes that we found in vivo and in vitro, we found that 263 of these genes were strongly upregulated only in the patients that have endocrine resistance. Importantly, when we go back to our in vitro model, these 263 genes are closing down when you don't have LSD1, and cJUN is not recruited anymore. And more interestingly, if you treat these cells with corin, these 263 genes are downregulated. So we propose that if there is a patient with endocrine resistance, if we can show that this patient expressed this, what we call CoREST signature, we could treat this patient with corin and maybe increase survival. So with this, I'm going to finish. So this is the summary of what I show you. So in sensitive cells, you have CoREST recruited to ER alpha and FOXA1 sites, which is important for activation of the estrogen responsive genes. During evolution of resistance, there's a switch, ER and FOXA1 are downregulated, and there's an upregulation of cJUN, which now is important for CoREST and BAF to activate EMT and metastatic genes. So this work is unpublished, is under review, and it was done by Liliana Garcia Martinez in the lab, a very talented PhD student. The Polycomb work has done by Eric and Candy, two former PhD students in the lab. These are our collaborators and our funding agency. And with this, I'm going to stop sharing, and I'm happy to take any questions. Thank you.
Thank you very much, Dr. Morey, for the excellent presentation. So cool to see all these new results.
Thank you.
We have two questions, and the first one is from Constantine Milonas. Does ring 1B knockdown impact breast cancer proliferation?
Yes, well, yes, we show that proliferation and cell fitness. Yes, a lot. Not only ring 1B, but also ring 1A. So we're investigating now what is the role of ring 1A also here, because we believe that ring 1A is the main E3 ligase here for estrogen ubiquitin. So both ring proteins are essential for proliferation and cell fitness, but through different mechanisms. And this is something that Andrew Adams in the lab, a new PhD student, is looking at. So yes.
Thank you very much. The next question is from Melanie Eckersley-Maslin. Very interesting talk. Do you have any insights into why it takes so long to get the phenotype switch in the breast cancer models? How many cell divisions have taken place?
Yeah, great question. A very difficult question, actually. About the cell divisions, we don't know. Lily had to spend 1 year going to the fax once a week to monitor this change and make stocks. So it was very time consuming. And now why? It's because the cells acquire this mutation in BRAF. We believe that the mutation in the MAP kinase pathway is what triggers this change. Now, why this is happening, we don't know. These are p53 mutant cells, so maybe they have genomic instability and they are more prone to acquire these mutations. but we don't really know exactly the mechanism. I would like to point it out that it's true in the lab, it took 1 year, but in the patients, it takes between 5 to 10 years. So in vivo, it even takes longer. And this is a very interesting question. And I don't know, I think it's very difficult to answer, especially in patients, right? Why patients need 5 to 10 years to acquire mutations in the estrogen receptor or in FOXA1 or yeah. Those are fundamental questions. Yeah. Very good question. Thank you.
Thank you very much. So is that one of the future directions of this project? Okay.
Yes, of course. Yeah.